Thanks, Andrew. So I'd like to give a quick report of latest progress on evaluating these nucleotide analogues and inhibitors of the SARS-CoV-2 polymerase. So thus far, we have experimentally identified 5 nucleotide analogues. 4 of them are FDA-approved drugs for other viral conditions as inhibitors of this new virus, COVID virus polymerase. So the sofosbuvir, tenofovir, and emtricitabine, AZT, and lofovudin. So these basically offer a sort of molecular basis that the molecules or with similar structure or their modified form can be quickly evaluated as potential therapeutics for COVID-19. So we heard in previous talk a lot of the genomics of this virus. This virus, from the molecular level to analyze, it is actually a very simple virus, much simpler than the flu virus. It has a single sort of a piece of RNA, positive RNA genome, and then it uses the RNA-dependent RNA polymerase to replicate in the host cell. So this is a RNA polymerase. It's thus well-defined and precise drug target because in human host, we don't use RNA-dependent RNA polymerase to replicate our genome. So as you heard from all the previous clinical trial report by Magda that a drug called Remdesivir is evaluated as a therapy for the COVID-19. Remdesivir is an inhibitor of this COVID polymerase inhibitors. It's not FDA-approved drug. It's currently in clinical trial. So there's our research program is targeted to use these nucleotide analogues to stop the polymerase reaction. Therefore, eventually the immune system can eradicate the virus. So our specialty is molecules. In particular, the design synthesis also study the structure activity of the molecules, nucleotide analogues, and the polymerase substrates. In this particular project, we're going to focus on evaluating these molecules and the polymerase terminators, because terminate polymerase determines the replication of the virus, eradicate the virus eventually. So important here, let's take a look at what it looks like in Remdesivir for the molecular level. So this is what it looks like. So the key part of this molecule is that the active part is going to be triphosphate. The other part are the regular type of nucleotide for since it's RNA, except the base here, you don't have a nitrogen group. And another modification is the cyano group here. So this has been experimentally shown to inhibit the polymerase of the SARS and MERS and so forth. And then we look at the other structures of similar molecules. For example, this one is sofosbuvir. It's being approved for hepatitis C. It's actually a cure for hepatitis C. Entirely eradicates the hepatitis C virus. So hepatitis C turns out has a similar replication system as the coronavirus because it's a positive strand RNA genome. So if you look at the structure of these molecules, have a lot of similarities with Remdesivir. Actually, the steric hindrance is much less. So therefore, we first want to evaluate to see whether this molecule can inhibit the new coronavirus polymerase. And similarly, we identified other molecules that have much less steric hindrance, AZT and so forth, evaluated as inhibitors polymerase. So here we collaborate with Dr. Robert Kirchhofer at the University of Wisconsin-Madison, who's expert in the polymerase of the coronavirus. So the prodrug sofosbuvir in the host cell turn into the active triphosphate, and this basically acts as a substrate for the polymerase. So we designed a simple template and primer based on this coronavirus genome, the new coronavirus genome, and use a rapid mass spec, multi-top mass spectrometry-based approach to evaluate whether these molecules are inhibitors of polymerase. And here's a template primer. Here, after the priming site, we build 2 A's, and then we add this nucleoside triphosphate because there are all U analogs. So this is the triphosphate of sofosbuvir. It's incorporate very well. So this primer peak, well, it's 2 A's. It's only 1 U incorporate and terminate, so further incorporation. So similarly, another molecule, 3' fluoro-TTP, also incorporate from a termination peak. And then similarly, AZT is a well-known anti-HIV drug, incorporate to the primer, and only 1 incorporate and terminate. So for these experiments, we use the polymerase from the SARS, the previous one. So this sort of established feasibility that the new coronavirus polymerase probably can also use this molecule as terminators, because we analyze the genomics, the polymerase genes of the SARS as new coronavirus polymerase is over 98% similar. Therefore, we select a group of molecules now. You can see these are prodrugs, FDA-approved drug, and here there's a triphosphate form. And then we evaluate all of them with new coronavirus polymerase in this simple polymerase biochemical assay, and it turns out all of them are terminators of the polymerase. It is sofosbuvir, shown here, incorporate a single one and terminate. It's almost 100% termination. You have very small primers. The next one, 3' fluoro-TTP, also incorporate and terminate really well. Almost all the primers are completely exhausted. The next, we look at this tenofovir. This is interesting, this molecule, you can see it's not a nucleoside per se, because it does not have a sugar. This is significant, but once incorporate, and then the polymerase, even they have the proof reading capabilities, cannot remove it because exonuclease cannot recognize this molecule. It turns out this molecule also incorporate and terminate because this molecule, tenofovir, acts as a analog. So therefore we need add 2 U's here. So 2 U's incorporate, and then the tenofovir incorporate from a termination peak. So next is AZT. It also terminate, but not as efficiently. We need to further optimize these reactions. So next we evaluate the combination of 2 molecules, tenofovir in combination with emtricitabine. These 2 are the components of the FDA-approved drug called Descovy and Truvada. They are the FDA-approved drug for treatment of HIV and as a chemo prevention of HIV. Essentially, they can act as a chemical vaccine for HIV prevention. And turns out both of these triphosphate form of these molecules also terminate this polymerase of the new coronavirus, as shown here. We use a similar assay to perform these kind of evaluations. So in summary, I think we at least identify 5 molecules that inhibit the polymerase of the new coronavirus. And again, this offers molecular level support that the molecules can be either modified or further evaluated the next step as potential therapies for COVID-19. And in particular for this Descovy and Truvada, because they're very successful use as a chemo prevention of HIV. If you demonstrate further activities of these combinations in the in vitro virus test in Lipkin and potentially move to clinical trial with Magnus group, and then this has the potential to be used as a chemo prevention of COVID-19. And finally, I'd like to thank other researchers, my laboratory working around the clock to generate data, and faculty collaborators at Columbia, at the University of Wisconsin, and Jack Ma Foundation for their financial support. Thank you very much.
Thank you, Jingyue. If there are any questions, I see there's a couple of people with raised a hand. Before we go to question, I wanted to ask one thing. I've just posted on the chat a link to a form that can be used to capture your contact information for all the participants in this webinar. If you fill that form, we will be able to keep you abreast of any new seminars and additional activities that relate to this form. So I encourage everybody to, of course, fill that information. Thank you very much. So we have a couple of questions here, if I can. Darcy, and John Rye. So Darcy, you can start.
My understanding of remdesivir is that it has some serious side effects. Is that also true for the anti-HIV compounds that are FDA approved?
So the FDA approved drug, the safety and so on and so forth has been established in terms of dosing, so on and so forth. The remdesivir, that's why it's still under clinical trial. And then for the molecular point of view analysis, you can see the difference, the remdesivir from all these molecules I presented, the FDA approved drug. The remdesivir has this cyano group, this CN triple bond. And if you're trained as a chemist, the metabolite of that cyano group, it's a very toxic molecule. So that's why there's some concern. You don't see many of the FDA approved drugs have this cyano group. So that's why it's under clinical trial, the safety need to be established, long-term safety.
Okay, there's also a question by John Riley. John, you can unmute yourself.
Hello, can you hear me? Hello?
Yeah.
Okay.
Yeah, we can hear you.
So because there's hundreds of thousands of people now on tenofovir as a prophylaxis against HIV infection, I'm just wondering, has anybody looked at incidence of infection in these people with the coronavirus?
That's a great question. Actually, we're under intense this discussion with experts in the field, like Professor Wafa Alsada at Columbia and Peter Anderson at the University of Colorado. They approached us. They say because a lot of people just say, right, it's under this therapy tenofovir. It'd be good to do an epidemiology survey to say whether they have low risk or they infected by the virus, and then what sort of progress of their disease. In a way, the data already there. If we can use kind of rapid survey approach and have a quick answer, that's the best clinical evidence, right? That's the molecules we identified might be potentially used as a chemopreventative care for the COVID-19.
Thank you. One last question, quick one from Martin Markowitz.
Oh yeah, hi. Thank you for your talk. I just wondered how quickly do you think that these drugs are going to go into the cell culture system, so you can demonstrate, see at what concentrations these drugs will demonstrate inhibition of the RNA-dependent RNA polymerase, because that's pretty critical, especially if you think about the drugs getting into the lung, et cetera, to really do what it needs to do in the cells in the lung.
That's right. So we're working around the clock with Professor Ian Lipkin's group and some others to move to the cell culture, and then eventually for clinical trial, we get evidence in the cell culture with the Magnus group. We thought that's why Columbia has all the expertise. We have a change events because the data you saw that's presented just like it's very new. We work around the clock, but we demonstrate at the molecular level, right? It inhibits the polymerase. Next step, we move to the virus. Next, with any model, and then the clinical trial. The good part of that, because all these molecules, we did not identify one, right? At least 5. We have multiple hits. 4 of them are FDA approved. In a way, I think have a concerted efforts, we might can get the answer really, really quick. That's why we work with multiple teams here at Columbia and all the other parts of the world to move this as rapidly as possible forward.
Okay. The only thing I will remind you is that in HIV, that there was no need to go from cell culture to animal models. It went from cell culture directly into humans.